Okay, welcome to the first afternoon session. And I think we'll start right off. Our first speaker is Ron Weiss from Princeton. Not to be mistaken with Randy Retberg, not that that would ever happen, but we're lucky to have Ron here to tell us about the iGEM competition that he's been involved in. And I want to make one other announcement. There's a slight schedules change, and Jay Kiesling will be speaking later tomorrow in the biofuels session, and Zach Serber is going to speak today in his place. So I think we have an exciting afternoon planned.
Okay, thank you. Okay, be on time, right? That's the key. Okay, well, thank you for inviting me. It's a pleasure to be here. As Pam mentioned, I'm not Randy. Randy has asked that I fill in for him and tell you a little bit about iGEM. So I will try my best. So Randy right now is in this building over here. I guess I don't know what time it is over there. 12? 1, 2? Yeah, he better be there because so he's working very hard on getting the Jamboree ready for you. And so he regrets that he cannot make it, but he hopes to see many of you over there at MIT. The other people that I need to mention that are actually responsible for iGEM, so I'm not trying to take any credit whatsoever here. So my former advisor, Tom Knight, I saw him before, I don't know where he is, came up with BioBricks and the notion of BioBricks, and there's also Drew, who's I think on a phone call right now. So he'll also make it to the end. If you have questions that I can't answer, he'll be able to also help you with those. So these are the people that are responsible for making iGEM happen. What is iGEM all about? Well, it is a friendly competition. We definitely want to mention that it's a friendly competition for undergraduate students. You heard today that actually there are high school students that are involved as well. And you saw 2 talks this morning that are very exciting. Show us what the possibilities are when you have people that don't know what's possible and what's not possible. So the deal is that these teams learn about synthetic biology. And then they come up with a project, hopefully some kind of cool creative project, and they talk about it during the Jamboree. So there's an emphasis certainly on educating the students to learn about synthetic biology, to be creative about synthetic biology, and definitely have fun. One thing that's really important, and they don't always necessarily notice this, is they fulfill some kind of a long-term goal, which is we don't know what this synthetic biology really is. We are trying to understand how to make synthetic biology a true engineering discipline. And by trying to educate new people, new members of our community on how to do this, we learn as well. And we're trying to understand what the principles are for making this a true engineering discipline that's going to be done in a reliable, predictable, efficient manner. And it's been quite fascinating to be involved in this competition and be involved also with the registry. And so a brief overview of where it's been. It started in 2003. It's a short winter course at MIT. I was actually involved as a guest lecturer at that time, organized by, again, Randy, Tom, and Drew. They did it again in 2004. Everybody had a blast doing it. And they decided to invite 4 other schools, and those include Boston University, Caltech, MIT was involved, Princeton, and UT Austin. And so we had a blast in the Jamboree, getting together. It grew to about 13 schools. Then we had 32, 54 last year. And this year we have about 84 teams participating in iGEM. And so you see that in terms of popularity, if that's the metric that you're going to go by, it's been a true winner. Okay, this is some kind of exponential growth. We have total participants that's also following the same trends. Soon, everybody in the world will be doing synthetic biology, and then we have to start. I guess then we create more life forms so we can sustain this, right? And so, but clearly, this is not the only means by which we judge how well this is going. People typically look at publications. So this is something. I'm not messing this up, right, Chris? This is something that actually was also involved with one of the original iGEMs, and Chris Boyd, Andy Ellington, and a group from UT Austin and UCSF were involved in making this happen. Recently, there's been a few other publications that have come up from undergraduate projects. This is in IET synthetic biology, an arsenic detection mechanism from EDMER, another one from Mississippi State for hydrogen detection. This is from University of Toronto. The idea here is that we get bacteria that are engineered to fluoresce in different colors based on the temperature. Another one from Brown. This is a tri-state toggle switch. And yet another one from Columbia, where we have another biosensor for iron detection. And so you see that these undergraduate projects actually can end up making it to publications. But I think more importantly, many of these projects that have been started by, again, by undergraduates that don't really know what's possible, don't know what the limits are, end up becoming flourishing into big projects in the labs where they got started, in the schools that got started, and they have long-lasting effects both in the labs and on other iGEM teams as well. And so as an observation about iGEM, trying to understand what's been going on since 2003, one thing that seems fairly obvious is the designs of the systems are certainly getting more elaborate, more creative. They're also integrating many different components. So we're not just talking about, for example, transcriptional regulation anymore. We're talking about incorporating many different facilities that are available to us in synthetic biology, looking at many kinds of applications, whether it's biosensing like I showed you, whether it's environmental applications, medical applications, trying to understand foundations for the technology. They're really sprouting to many different directions, and it's been quite exciting to be involved in that. Another metric is the increase in productivity. And so in 2004, in the first Jamboree, it was a lot of fun being in the audience, participating in that. But what we know is that people were excited, the students were excited to be there. But all the stories ended up being, we wanted to do this, we had all these cool ideas, but at the end, well, nothing really worked. And which is, emblematic of a lot of things in synthetic biology, at least in the beginning. And we said to ourselves, the question was, is this worth continuing? And, I don't think people really wanted to stop it. They said, well, at some point, things are going to come out of this, even in such a short time period. And it's just been amazing over the course of 2 to 3 years, the kinds of things that are possible, the number of parts the teams are able to synthesize, manufacture, and not just parts, but actually working parts, working composite parts. And now we go to the Jamboree, and there's some serious experimental results that are coming out from an amazingly short amount of time. And so the bar is being set higher and higher every year. And I think that's actually, even though we don't have a metric right now to show that, I think that's really indicative of the success of the program. And it's driving research both in iGEM, obviously, but also in synthetic biology. Something else that's being done is also the development of software tools. These are becoming more and more important as we venture to design more and more sophisticated systems. Where are we right now? So we're about to finish iGEM 2008, and this, again, is what Randy is doing, working on the final touches. Less than a month, we're going to have the Jamboree. 84 teams started. Most of them are still around. A number of people, over 1,000 participants from all around the world are involved, and about 850 are expected to attend the Jamboree. So that's a lot of people they're going to have to fit in the Stata Center. So I think they thought big in the Stata Center to begin with. They have room, but eventually that's going to run out of room for that. In terms of the kinds of projects that are going on, a lot of exciting projects. This is just about half of the projects. We'll see. I want to find out what the slot machine is all about. That sounds cool. A theme that's quite common to projects, I think, with students is some notion of killing and cell death. So we see the time bomb over here, kill bad E. coli. That sounds good. Search and destroy. So looking forward to what, I guess, Game of Life also has some notions of killing. I want to hear about the singing bacteria. I want to see what that's all about and see how they relate to the sound-sensitive bacteria. So lots of cool things going on there. Tell you a little, for those of you that are not familiar with iGEM, tell you a little bit about the structure, the schedule. So right after the Jamboree, we start round number, the next round. And so from November until next April, the idea is that teams get together, they learn about synthetic biology, they get funding for synthetic biology for their project. This is also a great way for, let's say, professors that are in schools that they're still not doing synthetic biology. This is a great way for them to become involved in the community. So this gives a great opportunity for learning. In May, once the teams have registered, DNA distribution takes place. last May, 2,000 parts were delivered to all the teams. Another thing that happens at the same time frame is a teacher's workshop. That's incredibly valuable. So staff, this is Tom and Randy over here, staff from iGEM go around and they actually help the teams that need it. I think everybody could use some help with respect to understanding how to run, how to set up their teams, and so on. So this is a great mechanism to get people involved, to get the education out there. And then I guess the fun part of the iGEM competition happens from June till November. So I guess this is the first time I've seen that it's really dictated. This is if you want to start working before June building plasmids, nobody is going to hunt you down, nobody's going to track you down. My guess is some people are doing that. This is the fun part. So you start all bright-eyed. You're going to conquer the world with this, and then you realize that there's some work involved. It's a little more complicated than programming Java or something like that. So there's some pain involved. Pain is good. You learn from that. But eventually, as we've seen in the last 2 years, definitely there's a lot of stories of success, and this is really the thing that's exciting about it. So eventually, people are happy again. So. Picture of the Jamboree. I didn't notice this before. I guess it's a blurry for effect. I'm not sure if Randy really intended for that. But it's the Jamboree is just exciting. There's so many young people moving around, trying to run from talk to talk, trying to catch all the talk, trying to find out what everybody is doing. Tremendous amount of energy at the Jamboree. Awards. So we give out awards. I guess we could say, from a politically correct perspective, awards are not what it's all about, right? I guess unless you win it, and then that's the only important thing in life. But certainly awards are at least show people what highlight some of the projects that people think are quite interesting. And then it's about building the community, right? It's about the fact that these are going to be the stars of the future for synthetic biology. Presumably there's nice overlap with the people here. These are the people that are learning about synthetic biology, but also teaching and understanding and figuring out how to do synthetic biology. And I think the overlap with the community here and the community at iGEM is really where the future is. So here's a schedule again. After the Jamboree, publishing your project sounds, you want to try to figure out how to continue working on the project. There are many cool projects that with some little bit of additional work could actually end up being publishable projects. So don't let go of those, after you're done with the Jamboree. Why? Why are we doing this? What was the rationale for starting this? And so the fundamental question, or one of the fundamental questions in synthetic biology, is can we use this notion of parts to actually build complex systems? And I guess by now everybody's heard about parts and parts and parts too many times. So I'm not going to belabor this point. Right? Is it the case that we can build anything we want by standardizing parts? Or is it the case that biology is too complex, everything is going to be a one-off? And so presumably, many of you are in this camp over here. A few of you heretics are in this camp right here. The answer is probably somewhere in between, right? It's not going to be as easy as we think to engineer biological systems. We're going to come up with standards, but we're going to have to think about what makes the biological substrate also not exactly like the other substrates that we're used to. We have to really appreciate the differences between them and make the appropriate changes and so on. And so we typically think of a computer, the revolution in computer, and the fact that parts in making these computers are known to us. We understand how to put them together. And that's the thing really that's enabled the entire computer revolution. And I like this analogy. The other thing that obviously has been very important here is the increase in the ability to synthesize faster and faster computers at smaller and smaller scales. And the analogy to that is the DNA synthesis that's actually exploding essentially at Moore's law kind of pace. And so it's really up to us to also understand what to do with all these capabilities, to understand how to design systems, how to be able to take all these parts and put them together so we can predict what happens. So TTL data book, this is inspiration for us, the notion that we have parts been characterized. We want to understand what the important features are for the biological systems and how to make them. We want to come up with the biological TTL data book so that we can understand how to engineer complex systems and actually predict ahead of time what might happen or what kind of parts we might need. Some of the parts, and so I'll just quickly, inverters people have seen, other parts that allow us to induce gene expression in various ways. And so the parts registry is there to collect them, right? But not just as a physical mechanism, but really as an informational mechanism as well, to understand what's important about the parts and also to try to develop mechanisms by which we understand how to put them together. And so the registry has just been a tremendous infrastructure for allowing us to coalesce over the important ideas in synthetic biology. In terms of the parts that are available, 2,000, as I mentioned before, have been shipped in the latest iGEM. They include basic parts. They include some composite parts. They include, it used to be the case that the focus has been on bacterial as a chassis. There's been more and more interest on other chassis as well, and I think we need to get an understanding of how to engineer those. What are the properties of this kind of a chassis, whether yeast, mammalian cells. There's also been some interest in cell-free chassis, and we'd like to be able to integrate that into the mix as well. So the iGEM philosophy, the get and give. We're going to provide parts for you. You're going to use those parts. You're going to rely on the fact that people have invested then blood and tears into making these parts, synthesize into characterizing it, into making them work, and then build more and more complex systems, more and more reliable systems as we move along, and then contribute back. Right? So the teams in the future will actually be able to benefit from all the hard work that you've been able to put in. This is an example of a part. That's a part for fluorescence, and you can see some description of the part with some experimental results. The registry does a lot, okay? And sometimes, as an outsider, I know that sometimes I ask the question, well, what do they do over there? Do they guarantee that all these 2,000 parts actually work well? Well, they do quality control, as Randy was telling me. So, for example, they verify the sequences. So they make sure that what is being given to them is actually what is being claimed. So they do sequence analysis, they do restriction mapping, they make sure that the antibiotics are actually working properly. A lot of work actually goes into this, and sometimes it's a little underappreciated. So send Randy your thanks, please. If if nothing else comes out of this talk, flood him with email, I guess, later on. What are the parts? So regulatory parts, for example, here we have a part where it has one input, one output, the notion of a promoter that's regulating the production of another gene on the outside. And so what we'd like to do, we'd like to put some kind of a black box around this where we have a notion of something coming in, notion of something coming out. If you were engineering this, then you want to know what's inside the box. But if you're trying to use this in a more composite sense, then you may not care about everything that's going on inside of here. Okay, so we'd like to find out what the interfaces are. Let me point out that this is not so simple, as most of you probably know. I would define this as one of the first grand challenge, perhaps, in iGEM, in the registry. What are the parts? What are the interfaces? Talk about, if we want to characterize the parts, if we want to understand how to be able to put them together, we have to, for example, characterize the steady state input-output behaviors, their dynamics, crosstalk with various other molecules that are going on. But some of the other things that are going on when you put a part into a chassis is what are the interactions? How is this part affected by the chassis? How does it affect the chassis as well? And so, for example, you normally wouldn't see these kinds of things being taken into account when you look at the TTL data book, but this is something we have to consider. Or maybe we can engineer some kind of a virtual system so that it's completely orthogonal to what's going on in the cell. Standard parts. Well, how many standard parts do we need? How many variants do we need to make in order for the systems to actually work properly? Right? These are important. There are many other important questions about the interfaces. What about parts that are not just about transcriptional regulation? If you look at the kind of parts that are out there, there's just a tremendous variety. How many of those do we have a good sense of how to characterize? Right? Whether it's things that are working at the RNA level, phosphorylation level, enzymes, structural proteins, and sensors. These are all parts that are being developed. How do we develop the interfaces? How do we develop the appropriate standards so that we can think about putting all of these elements together? Here's an example of a library of parts that from Chris Anderson's lab where they characterize many of the properties. For example, what are the expression levels from all of these parts? This is probably well known to many of you, characterizing a cell-cell communication device, trying to understand what are the input-output relationship, what are the timing, what kind of crosstalk. It'd be nice if we had this for every one of the 2,000 parts. This was a huge effort. This was, I think, at least a PhD, right? Still building on top of other work that's happened before then. How do we make this happen so that we have this much information everywhere? And so that's essentially grand challenge number 2. How do we characterize all these parts? We need to have functional characterization of all the things, maybe perhaps most of the things in the registry. I had a quick discussion with Randy about this. What's the appropriate way to do that? We don't know yet. Maybe possible to actually achieve this by coordinating the action from a few entities. Right? Thinking about what kind of incentive do people have for characterizing parts, for doing a good job at it, and then putting that information back into the repository. So maybe we need to actually figure out, let a few entities do the job, do the dirty work for us, let them benefit from it somehow, and then that helps the community as a whole be able to rely on all these parts that are available to them. Standardized measurements, let me skip over this for the sake of time. And this is essentially how things got started when Tom and I came up with this notion of the BioBrick standard assembly. Right, with this notion that we can take parts and take one part here, another part over here, digest them, and then make composite parts. And not have to think too much about how these parts fit together from the respect of being able to, needing to design PCR primers and things like that. So when you put two parts together, you have another part that can then be combined with other parts as well. So this is actually seems like a simple notion, but it's been a very powerful notion, and I think been tremendous. Had a lot to do with the success of the registry and the success of iGEM as well. And so at MIT, they're certainly trying to understand how to make this fully automated with robotic assembly. But then that comes to question the grand challenge number 3. So if we are able to come up with all these composite parts, we are able to design all these composite parts, what do we do? And I think this is actually more difficult. I don't think there's actually incredibly good ideas right now out there for understanding how to engineer systems at this level of complexity, higher level of complexity, that would actually work the first time or the second time or the third time. What are the appropriate system design principles? And I think iGEM is going to be one of the major ways by which we learn how to do this. Okay? So to summarize, can simple biological systems be composed from standard interchangeable parts? Perhaps. Cases where they are. Is biology too complex so everything is a one-off? Probably not. Answer might be somewhere in the middle here. And this is going to be critical for the success of synthetic biology. How am I doing on time here? Okay, so I'm pretty much done. So impact of iGEM is just so obvious that I'm not going to go into all the details. But it impacts students. Students learn. As I mentioned, instructors learn as well. This is a great opportunity for people to learn about synthetic biology, people that have been established in various other fields, and get into this field fresh and be able to contribute. So we want everyone to participate. Organize teams. Okay, organize teams for 2009. Join the registry. Contribute your parts. Use the standard notions of BioBricks and help raise money for the teams in your area so that you can move synthetic biology forward, move iGEM forward. And also, I want to make a quick announcement. Actually, this is relevant for this iGEM, that we actually need, because of the explosion in the number of teams, we actually need 10 more judges that will help judge the iGEM teams this Jamboree. So don't send me email about this. Send Randy some email about this, or Drew. They would love to hear from you. So these are the people that have been responsible for this. I just stood here and chatted for a little while. These are the people, the staff at iGEM Central, some other people, and Drew, Malcolm, and Jerry Sussman, who's actually one of my former advisors as well, so it's always good to say his name. People have been involved in iGEM, and certainly the funds have come from very important sources. So, and thank you again, Randy, Tom, and Drew, for making this happen. So I will stop there. Thank you.
Thanks, Ron, for conveying the excitement around iGEM. I'm sure there must be some questions. Yes, black shirt. You, black. whatever.
Got a microphone. First of all, I've got to congratulate you. This is really quite an innovative way of doing science and technology, and hopefully is going to be an important template for the future. I did have one simple question, though, and that is, we heard this morning from the UCSF team, which obviously engaged high schoolers rather than undergraduates. The question is that a one-off, or is there a place here for really engaging kids at a lower level in the system?
So I actually have an answer for that, which is I've been engaged with people from Boca Raton, from a high school there, who want to participate in iGEM. And I think that's going to happen. It's amazing what high schools have today. I don't think we want to go down to middle school. Maybe we should cut it off. There should be maybe kindergarten. I think, but I've had people in my lab that are not even allowed to drive yet, that are messing around with plasmids and lentiviruses and things like that. And they're fascinated. They know, they're sponges. They can pick up skills very quickly. They're very helpful in the lab, oftentimes better than the undergrads and the graduate students. So yes, hi.
As we saw.
Not my graduate students. My graduate students are
as we
awesome.
saw this morning. I think Drew wants to make a quick comment about this.
Ron, it seems like an outcome that you put forward was the research paper. But what I've been struck with is a lot of the iGEM projects seems like they could go out and have a much bigger impact on the world, right? So the arsenic sensor project from Edinburgh was a case where if that was partnered with Bill and Melinda Gates or somebody, right, you could see that taken forward and, if not commercialized, made available via some not-for-profit, a la what Amyris has done with artemisinin. And so I wonder if you've reflected or talked with other people about the idea of having a translational, development fund or a business competition associated with iGEM.
I have not. I mean, I certainly did not mean to have the publications as a metric. I was just showing that that's a standard academic metric, and from our jaded view, that's important. But certainly, I did not mean to imply that is the metric. And I think that what you've mentioned is important. Maybe we can team up for example, for the MIT 50K competition. I was actually involved in that back in '99 or something like that. And I think that would be something simple that we can do. Certainly, I'm sure there are other opportunities maybe in the Bay Area. There's one or two entrepreneurs that you can talk to. So yes, that would be very engaging. The question is, obviously, at what level, what's the readiness level?
I think also the mention of engaging foundations is also an interesting one as well.
Yes. The main question, or the big question of synthetic biology and iGEM in particular, is whether life and a living cell can be constructed starting from dead parts. And you mentioned something about escape rapidly from this question. Do you think this is a kind of iGEM project, build the living from parts, or is too early?
Well, so with respect to doing it in 10 weeks or the June to November, not quite yet. 7 days. Okay, I'm done. I think honestly, I think at this point, it's people are looking at cell-free extracts. There was a team. Remind me, the team that did the HL communication, was it from Imperial? I think last year was actually looking at cell-free systems, and certainly something to consider. I think if you look at synthetic life, synthetic genomes, it's still a little bit early, I feel, for understanding how to actually build something that operates, that has a programmable interface, which is so in terms of building lipid bilayers, in terms of things that can actually compartments that actually hold things, that's where the cutting edge research is right now with respect to synthetic life. So if iGEM teams want to engage in that, I think that would be great. But I think it's a little too early for them to actually now start thinking about putting all of these pieces together, the compartments and the maintenance of all the enzymatic and the regulatory activities within one cell. So that's going to have to wait until at least 2010 or.
I think later is Jeff Buca. Are you here somewhere? I think he's got a nice project that's not quite that, but he's engaged students in, where are you, Jeff? Anyway, I think he's giving a talk later about synthesizing yeast chromosomes by undergraduates. Where are you, Jeff?